Recording. Okay, guys, so let me pull the chat box up. All right. Can you all see my PowerPoint now? Okay, thank you. All right, so for today, we are going to mainly focus on chapter 1 that we went over, and then we'll start biomolecules, mainly getting into the vocab is what I really want to focus on because I know there was a lot of confusion there, and then carbs, and we'll start lipids. So first thing we need to know, and I'm going to try. If I draw on this, can you all see it? Somebody with a microphone, please answer if you don't care. Okay. So either nobody can see it or nobody has, we can see it. Thank you. It's okay if you all don't want to use your microphone. I can check the chat. That's no big deal. Okay, so let's start with life study to all levels. So you don't have a microphone. Oh, well, I'll have to play with that and see what I can do to get you all the microphone button. Thank you for letting me know. So I guess chat's what we got today. All right, so life studied at all levels. So we really will not focus on anything before cell. So the atoms, the molecules, the organelles, stuff like that. We don't really focus on that a lot. And the reason we start with a cell is that it's the smallest unit that's alive. So everything before that is considered, I'm going to introduce you all to a new word, maybe a new word. So if it's alive, that is called biotic. And I don't think we've introduced this term yet, but you will see it. So bio, meaning life itself, otic, the state of being. So this is the state of being alive. Now, if something is not alive, say like a rock, that would be abiotic. Simply adding that A prefix. Now, you guys will see this, I'm sure. I know we haven't covered it yet, I don't think. Ooh, that's not it. Let me erase that. Okay, so if something is alive, it is biotic. And if it's not alive, it's abiotic. And now we have the cell, which is the smallest unit that is alive. And then after that, we have tissue, and then organs and organism. These three are pretty much self-explanatory. The ones that I really would like to focus on are these two, because if any of them get confused, it is population and community. So population is a single group or species. So if we're talking about the population at UC, it would just be students or people. Now, in contrast to that, if we are talking about a community, it's multiple species in a place. So if we're talking about the people at UC, if we are talking about the community of UC, it would be students and squirrels. We're not the same species. We are obviously two different things, but we're in the same small area. We're still at UC. So if we wanted to expand this, it could be the trees and the students and the squirrels and the grass and I'm sure there are flowers here. All of those things. It's all biotic in a community. Now, an ecosystem, oh, here are these words. There we go. Biotic and abiotic. So an ecosystem is really focused on how they interact. So this is maybe how like water and let's do trees. Maybe if we're looking at that in an ecosystem. But it is all things in a particular place. So if we're looking at the ecosystem at UC, that would be the rocks and the road and the buildings and the trees and the students and the grass and the water and everything. Is everything in a particular place in a smaller environment. And then the biosphere is obviously the biggest, and it's just simply the earth. Bio, again, meaning alive, and then sphere, literally meaning circle. Okay? All right. So now hopping over to our themes. Starting off with evolution. I know she gave a steampunk moth example. Please make sure you understand that example. So I have it right here. Go back in your notes or go back and watch the video. Just please make sure that you are comfortable with that example. And if you're not, shoot me an email, make a tutoring appointment. Okay, so evolution. Sorry about that. I need to disable this. All right, so evolution is when current organisms are modified from the descent of a common ancestor. So this could be modification. So in our steampunk moth example, it was that after the steampunk era, white moths were easily seen by predators, so they would get eaten more. So then as a result, gray moths lived to reproduction age, made more babies, the population was mainly gray. So organism adapts to their environment. Obviously, the more fit you are or the more adapted to your environment, you have a better edge. This would be like taking a polar bear and putting it in the desert. It's not that adapted to that environment. It has a thick fur. It has other things that are adaptive to the cold. But hypothetically, through evolution, after generations and generations, another species would be produced that was adapted to that environment, hypothetically. So, let's see. Fitness and camouflages tend to be a factor in this. So back to our steampunk moth example, camouflage. This leads to the two main things of evolution. One, dead things don't make babies. And two, predators are lazy. So steampunk moth example. Predators won't go for the prey that are better camouflaged. They will go for the ones that they will stick out, less work for them. So in turn, the white moths will get eaten, the gray moths stay to reproductive age, leading to the second thing, dead things don't make babies. If most of the white moths are eaten, it stands to realize that the white moth population will slowly die out. Okay? This makes sense to everybody? And if you all have any questions, just pop them in the chat. And I'll switch back over. All right? Cell theory. So we've already talked about this a little bit with the levels of organization, but the cell is the lowest unit of life. So anything below a cell is not alive, like an organelle or an atom or anything like that. So all cells are enclosed by a membrane, which is kind of explanatory. You have to have I'm sorry, guys. Excuse me. You have to have an encasing on all the goo and the DNA and the organelles that are in the cell. They have to be in a nice little bundle, in a nice little ball. And then in a cell, they have to have DNA, which is used for genetic information. Yeah, just let me know at the end of the session. I'll leave a space open for questions. All right, so that leads us on to our third major theme, which is genetics. So chromosomes contain most of the genetic information. We will really start to focus not on this exam, but when we start the information for the next exam, chromosomes will be a focus. So you'll learn much more than you probably want to about that. So DNA is also the substance of genes. So let's put DNA to genes. We'll connect those two. And it has to do with inheritance and how traits are expressed. So inheritance and how traits are expressed. All right, so that's kind of an introduction to the three major things we will be learning about this semester. So now let's get into the parts of an experiment. So I will say that of this material, in my opinion, one of the hardest things is inductive versus deductive reasoning. I totally understand it is kind of confusing, and it's something that you've never heard before, or at least I had never heard it before when I came to this class. So I'm going to start with deductive because I think that's kind of more intuitive. So deductive, detective. So it's using pieces of evidence to come to I'll put down here one logical conclusion. So this is the funnel. So you have all this evidence, and then you have one conclusion. So I don't know about you all, but I am kind of obsessed with crime shows. That is why I say deductive, detective. So in a situation like this, say the evidence could be fingerprints or DNA or maybe video evidence. So if we see a video of Bob murdering Susie, the one logical conclusion is that Bob killed Susie. There is a lot of evidence. Say there's fingerprints, there's lack of alibi, there's presence of motive, there's the video. There's a lot of evidence, and it comes to one conclusion, one solid conclusion. Okay, now the flip side of this is inductive. So this is where you make a broad, so you flip the funnel upside down. So many observations. So observations. That's not how you spell observations. All right, so many observations make a generalization. So say a broad conclusion. So an example of this would say, let's say I tested a bacteria and it is green and fuzzy, and I determined that that bacteria is called, I'm going to make up a bacteria name. Let's say fuzzy. We're just going to name that bacteria fuzzy 1, okay? So a generalization would be, because this bacteria is green and fuzzy, and the name of this is fuzzy 1, then all green and fuzzy bacteria is this particular species. On the flip side of that, if you don't like bacteria, that could be saying that my dog is black and white, so all dogs are black and white. Okay, that's taking a few observations and making a very broad conclusion or a very broad generalization. Okay, any questions about this? Okay. Now we're going to move on to parts of the experiment. So firstly, whenever we make an experiment, we want to make sure that it is controlled. Controlled in the sense that we have something to compare it to. So if we want to say that fertilizer will make this plant grow taller. All right, so say that this is my fertilizer. Added. Okay, cool, we've done the experiment. How do we know if it grew taller than if we didn't add fertilizer? We have to have something to compare it to. So say we go here and say this is my plant effort. So now we can very clearly see that fertilizer made this plant grow taller. Now, on the flip side of that, we would also have to keep everything exactly the same between these two groups to make sure that it's a right comparison. That could be such as the type of plant. We want to make sure, say these are both bean plants. I want to make sure they're both the same type of plant, that they both get the same amount of water. So they get the same amount of water. That they both have the same amount of sunlight, or XYZ, that they are both in the same conditions. The only thing that's changed is fertilizer. Now, in building off of that, in this case, fertilizer would be the independent variable. It's what you are changing. I can change it. So as a scientist for this experiment, I can change if there's fertilizer or if there's no fertilizer. So I can add fertilizer to this one. I cannot add it to this one. I can change it. Now the dependent variable is just what is being measured. So the dependent variable is kind of like your results. So in this case, it would be plant length or stem length. I think that's all right. Okay, now one more thing. Say we were making a graph. Say we were making a graph of our results. The independent variable always goes on the x-axis, and the dependent variable always goes on the y-axis. Okay? All right. Now moving on to hypothesis, theory, and law. My best advice for this is to word associate everything. So a theory is broad. A theory is the biggest one. A broad scope and is backed up with a lot of evidence. On the flip side of that, a law is generalized. And it is based on repeated tests. So still a lot of evidence, but not as much as a theory, per se, has. So a law is kind of general. Now, a hypothesis is a single if-then statement. single if-then statement. And as we learned in class, what are two things that a hypothesis has to be? It has to be testable. I'll give you one. Can anybody tell me the other one? Falsifiable, exactly. Good job. It has to be testable and falsifiable. Okay. So that's all I have on chapter 1. So now let's switch over to biomolecules. But before I do so, are there any questions on the chapter 1 material? Okay. So the best thing that we need to do for this chapter in biomolecules is to learn our vocab. So let's break this down. Let me change colors here. All right, so hydro is seen on both of these words, hydrophobic, hydrophilic. And we know that hydro means water. So on the flip side of this, let's think hydrophobic first. We've all heard phobia, phobic. So it's fear, dislike, anything of that sense. So hydrophobic literally means fear of water. So it will repel water, it will not mix, it doesn't like water. So when you think of not mixing with water, what's the first thing that you would think of? What doesn't mix with water? Oil. Good. And we will learn later that oil is a type of lipid. Okay, so now let's go to hydrophilic. So philic means like or loving. So this literally hydrophilic means water loving. So this can be, it would mix. It would mix with water. So something that does mix with water would be like sugar and table sugar, and that is a carb. So carbs mix with water. Lipids do not mix with water. So that's what we're meaning when we say hydrophobic, hydrophilic. Okay, so hydrolysis and dehydration synthesis, I have slides dedicated to this in a second that we'll go over. Hydrolysis is breaking a bond, so let's just look at this. Hydro, again, means water, so we know it has something to do with water. And lysis literally means to break. So hydrolysis quite literally translates to water breaking. So adding water to break a bond. Okay. And then dehydration synthesis. Dehydration, we know, means to take water away. We've all heard of dehydrated if you don't drink enough water. And synthesis, I know we've all heard of this, is to make. So this is literally taking away water to make something. And I have slides dedicated to this, so we'll go over it. Okay, so next we need monomer and polymer. So a monomer is just one single molecule. So it would be like, it would be like one single carb. That's a monomer. Let me add my OHs, make it a carb. Okay, one single carb. Polymer would be, please ignore my horrible drawings. multiples, so we have 3. Now, a lot of people in my Socratic were confused. Monomer means 1. Di means 2. Poly means just more than 3. Or actually more than 2. So 3 or more. So this would not be a trimer. Trimer? This would just be a polymer. Okay? All right, so dehydration synthesis. That's when you take 2 separate and independent molecules and you make them 1. So I know that she used the Janet and Carl example in class. I like to give you all an alternative and just draw it out and see. Sometimes the analogies can overcomplicate it for some people. So let's just draw this out and see if that might help you a little bit more. So we have 1 OH, and I won't draw it, but we'll make this a carb so it has a lot of OHs and a carbonyl group on it. And now you have this other one. Now, we know dehydration is going to expel a water. So water is expelled. Now, we know that water is H2O. So we have 2 hydrogens, 1 oxygen that is being expelled. And we know that the bond between 2 molecules are held by oxygen. If you look up here, and oxygen links them. Okay. So, let me change my colors here. I want to take water away. So here we have 2 H's, 2 O's. We're going to get rid of that water. Now, all we have left is that 1 oxygen. That is horrible. And because those are now linked, because we wanted to take 2 monomers, 2 separate, and now these are linked. Now this is 1 big molecule. Does that make a little bit more sense if you were struggling with this? And there is a worksheet, I believe on week 3, that is not for a grade, it's just for practice, but I highly recommend if you're struggling with this, try to do it and take it to a tutor to check it, or if you look at it and you're like, I'm so confused, take it to a tutor. We will sit down and work on it with you, but you need to really understand this, okay? All right, so on the flip side of this, hydrolysis, we already said hydro means water, lysis means break. So this is breaking with water. So water is added. Now, I'm going to draw up here. Water is H2O, 2 hydrogen, 1 oxygen. Okay? So let's take our same molecule that we had from last time. We'll pretend it looks the same. I know my drawing is not wonderful. So let's pretend it looks the same. We have this little linking oxygen, but we want to get them separate. So we want to add in H2O. All right, so our goal is to separate these. So let's go ahead and draw them separately. Connect that. All right, so there's an oxygen already here. So I'll just make it on this little guy. And then to make that hydroxyl group that we need to put back on there, we're just going to put a little hydrogen. So now we only have 1 hydrogen left. Now we have 1 oxygen that needs used and that other hydrogen. So the basis of this is that most linking is going to be associated with hydroxyl groups, which is a functional group that is on the next slide, but it is basically just OH. That is a hydroxyl. And again, if you break that word down, you have hydrogen, and then oxyl, oxygen. So it's literally an oxygen and a hydrogen. And then once you take out H2O, you just have this linking bond. There's that one oxygen left to hold them together. Okay, that is what happens in dehydration synthesis. In hydrolysis, you just have this linking bond, but water is going to come in. It's going to break that bond. I like to say that there's no room once you add water in, so the bond just breaks, and now you have two hydroxyl groups. Does this make more sense? Okay, taking that as a yes. All right, so functional groups. Before we even start to kind of learn the different types of biomolecules, you guys need to know how to identify them. So we already know about our hydroxyl groups. It's just a little OH. Now, a carbonyl group is C double bonded to an O. That's what will mainly show up on a molecule. It also has this R group, which just means other stuff, and a hydrogen. But what you really need to look for is C double bonded to an O with really nothing else on that carbon. So, say if I wanted to draw a carbonyl on this here molecule. Now, remember, every crosshair is a carbon. Okay, so if I wanted to do that, I would just draw a double bond from that carbon, put an oxygen. I know that looks really sloppy. Okay, that is called a carbonyl. Now, very similar sounding is carboxyl. Now, let me change colors here. And if we look, we see something here that we've seen before. That's a hydroxyl group. So how I remember this is the carboxyl is kind of like the baby of these two. Because if you look, we can break it down to it has a hydroxyl group. So you have that carbon, and it has a hydroxyl group. And then it has this C double bonded to an oxygen. So it kind of has properties of each of the other functional groups. I've also had kids say that oxyl, it has the extra oxygen. So it has two oxygens here where this one only has one. I have heard kids say if it has the hydroxyl and there's more stuff on it, then it's the carboxyl. Kind of the most simplest, if you know your stuff, is just to look, find your center carbon, and then see if there's other stuff on it. So a carbonyl is just that double bonded oxygen. A carboxyl has the double bonded oxygen and an OH group on it. Okay? Any questions about that? Okay? So now we're going to hop on over to carbohydrates. So carbohydrates are used for fuel and building blocks. So what you can mainly identify a carbohydrate with is that it has a carbonyl group and lots of OH groups. So we'll come over here and let's pick out that carbonyl group. We're going to pick it out right here. It's got that C double bonded to an O, right? All right, so check, it's got that. Now let's look for the OH groups. Here's an OH group, here's an O. Okay, that's a lot of OH groups. So that's how I would check to see if that is a carbohydrate. Now, this is just a little equation I thought would be helpful. 2 sugars minus water equals 1 big sugar. 1 big sugar plus water equals 2 big sugars. Just to kind of get in your head how to apply dehydration synthesis and hydrolysis. So carbohydrates and water. So let me draw H2O up here for you all. So we have H2O, and what that would look like would be an oxygen with 2 H's hanging off of it, okay? Now, if we look at this molecule here looks very similar. Let me change colors. Looks very similar to an OH, right? So I remember this like a lot of OH groups mix well with water because they look similar, okay? And if you just want to put their little equations up here, that also looks similar. H2O, OH. Very similar. So they mix well together. That makes them hydrophilic, water-loving. So the main thing when you're trying to determine if they are polar, so water is polar, OH is polar. If a molecule is polar, that makes it hydrophobic. Look for the oxygen. If there are a lot of oxygen on a molecule, it is polar. Now, I say to specifically look for the OH groups on carbs because they are the most distinguishing form of oxygen you will see on a carb. Okay, does that make sense? All right. Now, lipids. A lot of hydrocarbons. Now, I didn't put this in our functional groups, but a hydrocarbon is just a C with a bunch of H's hanging off of it. So literally hydro, hydrogen, carbons, carbons. Now, a lipid has a carboxyl group. Remember that carboxyl was the carbon that has a double bonded oxygen and a hydroxyl group. So we have this right here. Maybe. Ooh, what happened there? Okay. So we have that. Right. Whew. Sorry, I can't talk apparently. So we have this carboxyl group, this string of hydrocarbons, hydrogen, hydrogen, all on a bunch of carbons. And now another distinguishing factor is not a lot of oxygen. Now, do not look at a molecule and be like, oh, I see 2 oxygens. That means it's a carbohydrate. Don't look at it that way. Look at it in the sense that every single lipid will have to have at least 2 oxygens because it has a carboxyl group, which has 2 oxygens. Look at the rest of it. You can't pick out a lot of the oxygens like you could here. Here we had oxygen, 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 a lot of oxygen. Here, there's none. But if we change this guy to an OH, that wouldn't change the structure because there's just one. There's 1, 2, 3 compared to this whole big line of carbons. So all lipids will have some oxygen. It's just the amount. They won't have a lot. All right, so how do you think, let's flip this up here. If carbs liked water because they looked like them, because they had those hydroxyl groups, how do you think that lipids would work with water? Remember, they don't have a lot of those hydroxyl groups. They don't have a lot of oxygen. How do you think those would mesh? How would lipids and water mesh? Would they mix or would they not mix? Not mix, exactly. So would that make lipids hydrophobic or hydrophilic? Hydrophobic. Good job. Good job. All right, so now switching over. Lipids don't have a lot of oxygen. They have hydrocarbons. Hydrocarbons. None of that word sounds like oxygen. Hydrocarbons. So they have very little oxygen, which makes it nonpolar. So a nonpolar molecule, so water is polar. So if a lipid is nonpolar, that means that a nonpolar is not going to mix with something that's polar, which means it is hydrophobic. Water fearing. And this is kind of intuitive. You can think oil and water. They don't really mix. Or if you melted some butter, it would float on top of water. So lipids are hydrophobic. All right, I think, correct me if I'm wrong, is this where we stopped in lecture? Yeah. Okay, guys. So, as always, I will send out this PowerPoint. I do have a question for you all. I can save this PowerPoint with what I've highlighted and written on it and send it to you, or I can send it like a clean version of this to you. What would you all prefer? Let me stop sharing my screen. Extra notes. Okay. All right. Sounds good. I will send that out. I just got a new laptop for Christmas, so I'm playing with being able to actually write on a slide. Very helpful. Anyway, so is there any questions about this material? If not, I'll stop the recording, and if you have questions about lab or any homework or anything, then I'll hang out for a few minutes after. So any questions about this material? Oh, so sorry, I have to take a picture of you all, so the name, so I can take attendance and you can get your bonus points. Okay, that would have been bad. All right, so now that I've taken a picture, if you don't have any questions, you guys are free to go. If you do have questions, I will hang out for a few minutes, okay? The exam, I believe it is 2 weeks from Friday. So you have next week, full week of lecture, and then the next week, the exam opens on Friday.